Yes, good morning, students. So we should begin with our new practical today. So our new practical is the study of the animals. Let me start with it. So fine. So let's start with the biology practical for the class 11. So this is another practical in the row. Today's aim for the practical is to study the animal specimens, and we have to correctly identify them on the basis of their diagnostic features. Fine, that we're going to study today. So what are the material required? We require the specimens, virtual images for it. Fine. So let's begin. So first specimen for today's study is the sponges. Fine. The sponges, they belong to the phylum Porifera. Fine. So these sponges, they are mostly marine, means they are found in the sea water. But some sponges, they are also found in the fresh water, such as the ponds and lakes. Example is the spongilla. So you can see this is spongilla growing in the freshwater ponds and lakes. Fine. The size of the sponges, it varies uh, simply from 1 centimeter to about the 1 meter in length. Fine. So some of them, they are irregularly shaped like this, but some they are vase shaped like this. See, all these sponges, they are multicellular, means their body, they are made up of many cells, and they only have the cellular level of organization in their body. Means their body only consists of cells only, no tissue, organ and organ system level of organization is found in them. Only cellular organization is found in them. So in their body, only cells are found. They are active, they are independent, they carry out all the functions in their body, and they can be arranged as only as a loose cell aggregates. So these sponges, they are irregular in shape. That's why they are asymmetrical, because we cannot cut them into the equal halves because their body is irregular. Fine. So most important feature of these sponges is that they have the canal system. They have the water transport system in them, which is very helpful for these sponges for the nutrition, for the purpose of nutrition in them. Food simply passes along with the water current inside their body, and they will simply trap the food, digest it, and absorb the nutrients. So, and this water transport system is also essential for the reproduction purposes also. Gametes also come along with the water current inside their body. Now, these sponges, they reproduce by the means of the asexual reproduction as well as by the sexual reproduction. Asexual reproduction, it occurs by the means of the budding. In the process of budding, what will happen? A small bud arises on the parent plant. This bud, it grows, finally detaches from the parent plant, and this bud will grow into a new animal. Fine. So new sponge will be there. Fine. So now, in the case of sexual reproduction, they involve the two sexes, male and female sex. They will produce the two different gametes, female gamete ova, male gamete spermatozoa. The fusion of these two takes place, zygote will be formed, and this zygote will develop into a new animal then. Fine. So in this way, their life cycle will complete. Fine. So we don't have model for it. We only use the virtual images for its identification in it. So now let's move on to the next specimen. Fine. So now the specimen second is a hydra. So this is the classification for this hydra. Kingdom is the Animalia, phylum is the Coelenterata or Cnidaria, and the class is the Hydrozoa, order is the Hydroida, and type is the Hydra vulgaris. Hydra vulgaris. Fine. So this is a scientific name for this hydra. So here you can see this hydra, it is a freshwater animal. You can find it only in the freshwater ponds, lakes, streams and rivers. Most of the time it remains attached to the substratum like that of the rock and soil with the help of its base. This is the base. But sometimes when it detaches from its substratum, it simply swims in the water. It somersaults in the water. Fine. So with the help of this tentacle, it simply swims in the water. Now, here you can see the body. When we see into the body of this hydra, the body of the hydra is cylindrical. It is tube-like. It is tube-like with a closed base at one end. So this is the basal end with the help of which remain attached to the substratum. And a single opening is there at the anterior end, at the oral end. This is called as the mouth. This is called as the mouth. So this is the mouth which is situated at the anterior end. Terminal opening is there, which is called as a mouth. So in these hydra, only a single opening is there, only mouth is there, no anus is there. Fine. So this only single opening will be there. So this mouth, it remains surrounded by the means of a dilated region, which is called as a hypostome. This hypostome, it remains surrounded, it bears like 6 to 10 finger-like, thread-like projections. These hollow projections, these are called as the tentacles. So these are the tentacles. So in other words, what I can say, the mouth, it is situated at the anterior end. It remains surrounded by the means of these contractile tentacles. They are very contractile in nature. With the help of these tentacles, this hydra can move, can locomote, can capture the food, can capture the prey for itself. Now the body wall. So the body wall of this hydra, it is mainly made up of two layers. Two layers. That's why this animal, it is diploblastic, because its body is mainly made up of the two layers. Outer layer is called as the epidermis and the inner layer is called as the endodermis. And in between these two layers, only a mesoglea is present. A jelly-like substance is present. Mesoderm is entirely absent. So that's why this animal is diploblastic only. Now the epidermis, the outer layer of this body wall, it mainly contains some stinging cells or nidocytes or the nidoblast in them. Here you can see epidermis, here you can see the nidoblast, nematocytes are there, nidocytes are there. So it is like that structure. Here you can see it is the structure is like that. Fine. Cup-like structure is there. Inside it, a thread is there. A coiled thread is there. Fine. So when it is in the resting stage, this coil, it remains inside this cup-like structure. But when some predator approaches to this, or when this hydra wants to feed upon its prey, so now at that time, what it will do, it will simply retract. It simply retracts or throws its thread, coil thread, it moves its thread out, it throws its thread on its prey and it captures its prey, it paralyzes its prey and simply eats it up. Same mechanism goes for the predator. Predator के case में what will happen? The predator will go away. Predator will simply go away because it gets paralyzed by these nematocytes, by these needoblasts. So it is simply, so it can be used as an organ of feeding also, or it can be used as an organ of defense or offense also when some predator approaches to it. Now, the reproduction. So the reproduction, it occurs by both means. Asexual reproduction by the means of budding. Here you can see a small bud is coming out. This bud detaches from the parent plant and grows into a new hydra. And sexual reproduction is also found by the means of the gametes. These gametes, they are produced in the gonads. These gonads, they are present in the body wall. Fine. So in this way, the sexual reproduction is also found. So we have a model of it. We have a model of it in our lab. So this is the model of the hydra. Here you can see the hydra is there. The whole body of the hydra is there. Fine. So now this is the basal end. This is the terminal mouth. These are the tentacles. These are the tentacles, and this is the small bud which is coming out. Fine. So this is the model of the hydra. Fine. So what are the diagnostic features on the basis of you have identified cylindrical body, mouth surrounded by the means of the 6 to 10 tentacles. So in this way we have identified it. It is a hydra. Now moving on to the next specimen. So our next specimen in the row is it is the specimen Fasciola hepatica. Commonly it is called as the liver fluke. Liver fluke. This is the classification: kingdom Animalia, phylum Platyhelminths, class Trematoda, order Achinostomida, type is the Fasciola hepatica, or commonly liver fluke. Fine. So this liver fluke actually it is an endoparasite. Endoparasite means it lives inside the body of some host, some host. So in this case, the host is these are the sheep. It can be goat, it can be cattle like cow and buffalo, and it can be human also. So it lives inside the bile ducts, in the bile ducts of the liver of these all these animals, including the human also. Fine, where it causes a severe liver disease in these hosts. This disease is called as the liver rot. Liver rot. Fine. So when we see into the structure of this fasciola, it is triangular in shape. It is look like a leaf. It look like a dorsiventral flattened leaf-like structure. So it's a parasite which lives inside the body of these hosts. The host can be anyone. It is about the 25 mm in length. It's so small. It's 25 mm in length only. So this is the body of this fasciola. So here you can see a blade-like, a leaf-like, flat triangular shaped body it has. So this is the anterior end, this is the posterior end. At the anterior end, a single opening is there which is called as a mouth. This mouth is remains surrounded by the means of an oral sucker, with the help of which it remains attached to the bile ducts of the liver inside the host body. And another sucker is also there. This is called as a ventral sucker or the acetabulum. Acetabulum. Acetabulum sucker is also there, with the help of which it also remains attached to the bile duct of the liver. Now, the whole body of this fasciola it remains covered by the means of the cuticle. Cuticle is there. Fine. And this animal actually it is a hermaphrodite animal, means both the sexes, male and female sex organ, they are present inside the same animal. Inside the same animal, fine. So, and it also has a common genital aperture. Here you can see the common genital aperture or the genital pore is there, in front of this acetabulum or the ventral sucker. Here you can see. And since it is a host, so its life cycle, it requires two hosts to complete its life cycle. So such type of life cycle, it is called as a digenetic life cycle because it is completed in two hosts. The primary host is the man, or it can be the sheep also, goat also, cattle also. And secondary host is a snail. It is a non-chordate, it is a snail. So these two hosts, they are required for the completion of its life cycle. So now we don't have the specimen of it, only the virtual images we will see. So how you will identify it? It is a leaf-like flattened structure is there, which is called as a fasciola. Fasciola. Whole body is covered by the means of the cuticle, and two suckers are there. One oral sucker surrounding the mouth, and one ventral sucker. So on the basis of it, we have identified it as a fasciola. Now moving on to the next specimen. Now the specimen 4. So now the specimen 4 is the ascaris. So now here you can see the ascaris lumbricoides or the roundworm is there. Fine. So now this is the classification. Kingdom is the Animalia. Phylum is the Esculminthes or the Nimehelminthes. Order is the Ascaroidea and type is the Ascaris lumbricoides. Fine. Again, it's an endoparasite. It is an intestinal parasite which lives in the intestine of the humans, especially the children. Fine. So this is the structure of this Ascaris. It has a cylindrical body, cylindrical worm-like body with a tapering end. Both the ends are tapering, sharp. The anterior end, this is the anterior end of this. This is the anterior end. At the anterior end, the mouth is situated, and at the posterior end, the anus is situated. So such type of digestive system, it is called as a complete digestive system having the two openings. That is a mouth, terminal mouth, and a posterior anus is there. Fine. So this mouth, it remains surrounded by the means of a three lips. So it has a tri-radiate mouth in it, and tri-radiate mouth at that terminal end, and just beneath this mouth an excretory pore is situated. Excretory pore is situated just behind this anterior end, which is containing the mouth. Sexual dimorphism is very distinct in this ascaris. Here you can see both the sexes, they are different, they are distinct. You can easily identify. And males, they are small as compared to the females. Females, they are very big and larger. And the males, the posterior end of this male, it is coiled and it is look like a J structure, like a J alphabet. So it is a male. The cloaca is there, and inside this cloaca the pineal spicules or the seta are there, which is projecting out from the anus, and with the help of it, it simply copulates with the female. So it is used in the purpose of copulation. Fine. So in this way, you can see a clear distinction is there between the male and female. See, this is an endoparasite. It is an endoparasite which is living inside our intestine. So it causes a disease. This disease is called as the ascarisis. Ascarisis. In ascarisis, what will happen? These ascaris, our intestine it gets filled with the large number of these ascaris, which results in the obstruction of our intestine. Obstruction of our intestine. Such person which are suffering from this disease, ascarisis, what will happen? They will feel difficulty, discomfort in their intestine. They feel nausea, they feel vomiting, they have high fever. So what is the method? What is the remedy? The remedy is that they require the surgery. They require the surgery in which all these ascarises which are filled in their intestine, they are simply taken out from the body of that patient. And some in case of the low grade infection, medicines can also work. So with the help of the medicine, these ascarises they also comes out from the body along with the feces of that patient. So in this way, how you have identified? This is the male with a coil, J-shaped posterior end, and this is the female, big and large, cylindrical body, whole body covered by the means of the cuticle. So in this way, we have identified it. Now the uh, next specimen is a ferritima. Ferritima posthuma is commonly we call them as a earthworm. Kingdom is the Animalia, phylum is the Annelida, class is the Oligochaeta, order is the Terricolae, and type is a Ferritima posthuma, or commonly we called as the earthworm. So these earthworm you must have seen. They live in the burrows in the moist soils, and during the rainy season, like now, they simply come out of their burrows. It has a long cylindrical tube-like body. It has a segmented body like this. It measures about 10 to 20 centimeter in length. It is quite slimy in touching, both slippery and slimy in touch, and they are somewhat pinkish to brown in color, like that of the soil. And the mouth, here you can see the two ends are there. The two ends are there. This is the anterior end and this is the posterior end. At the anterior end, mouth is situated. At the posterior end, anus is situated. Again, our complete digestive system is there with two openings, mouth and anus. Now, here you can see, we can easily identify the dorsal and ventral surface of this earthworm. How? The dorsal surface is somewhat dark in color and it has a clitellum band on its dorsal side. And on the ventral side, you can see the genital apertures you can see. Genital apertures or the gonopores you can see. These gonopores, they are present on the ventral side, which is somewhat light in color. Somewhat light in color as compared to the dorsal surface. Fine. So darker dorsal surface, ventral surface is lighter, having the genital pores. And the segments 14, 15 and 16, together they fuse together to form a band. This band is called as the clitellum. This band here you can see it is somewhat thick band. It is called as the clitellum, which is present there on the body of this earthworm. And the whole body of the earthworm, it is metamerically divided into the segments. And these segments, they are called as the metameric. And each of these bears a yellow tiny setae you can see, the paired setae you can see. These look like the tiny bristles that are present on these segments, each of these segments except first and last segment. All these segments they bear these paired setae with the help of which This earthworm, it locomotes, it moves from one place to another. So these are the organ of locomotion seated in these earthworm. And what is the function of this clitellum? See, this clitellum, again this earthworm, it is a hermaphrodite animal. It is a hermaphrodite animal, but cross pollination is the rule. It undergoes cross fertilization, sorry, it undergoes cross fertilization in it. So this clitellum actually it produces or more cocoons one or more cocoons or the egg case in which the ovas they are laid down, which are filled with the ova, and later these ovas will get fertilized by the sperms from the another earthworm. So in this way they ensures cross fertilization in them, in spite of being the hermaphrodite animal. Fine. So this all description is for the earthworm. So how you identify it? It is a long cylinder segmented body having the both dorsal and ventral surfaces, dorsal darker, ventral lighter, having the genital apertures. And the most prominent diagnostic feature is that the prostomium is there. It is which is surrounding the mouth opening, and a clitellum is there, which is a band-like structure, which is present at the 14, 15, and 16 segment of this body of this earthworm. So these are the diagnostic features on the basis of which we have identified this earthworm. Fine. So now, let's look on to the how the specimens we have. We have specimens. So now let's look. So we have the specimens for earthworm. So here you can see this is the specimen. This is the specimen for the earthworm. So this is the earthworm which belongs to the phylum Annelida. Here you can see a cylindrical pinkish brownish body you can see, and whole body is divided into the different segments, and seta are there for the purpose of locomotion. Mouth is there at the anterior end, anus is there at the posterior end. So this is the earthworm. We also have a model for we also have the specimen for ascariasis. So this is the ascariasis. So here you can see the ascaris is there. The coiled part, it is the coiled part. So definitely it will be a male, and it is shorter in length. And here you can see the another specimen is there. It is larger, it is much bigger, and it is having no coiled or J-shaped structure in its posterior end. So certainly, most probably, correctly, it will be the female ascaris. This is the male ascaris. So we have the specimens of these in our lab. So this is all about for the part 1 of study of the animal specimen. So we will be continuing with the part 2 of this animal specimen in our next practical. So stay home, stay safe. Bye. Have a great day. Bye.